everyone. Welcome to your Chapter 17 video. This week's announcements. Let me go here to my Draw tab, make sure that my pen is functioning. Let's go to black. Okay, so this week's announcements. There will only be videos for Chapter 17 this week. As of right now, I have planned 2 videos. Okay, this one that you're watching, and this one is going to cover transcription, and then a second one that covers translation. Now, because these are such important and central concepts to biology, we're only going to cover Chapter 17. I really want to make sure that you get these concepts down pat. That means that you do have a quiz this Friday. So let's see, today is the Tuesday the 27th, so 28th, 29th, 30th. So I think Friday is the 30th. Of course, of October. So you do have a quiz this Friday. As usual, it will occur at the beginning of class during your scheduled class time. And of course, as usual, all of this only applies if you are watching this video in the fall of 2020. But your quiz will cover just Chapter 17 only, all right? So what is Chapter 17 all about? Well, as I mentioned, it is going to cover both transcription and translation. And I referenced that a little bit in our Chapter 16 lecture video. But let's kind of go back and refresh our memories as to where we are. So remember that the central dogma of biology states that DNA is transcribed into RNA, and that this RNA serves as a message to become translated into proteins, right? And then you all understand, I hope, that because proteins are essentially the workers of the cell, it is the action of these proteins that ultimately result in phenotypes or traits or however you want to think about that. But the physical things that we observe, those are all the results of the actions of proteins in your cell, okay? So we're going to focus in on these 2 processes and how they happen at the molecular level. Now we're going to start with this particular video in thinking about transcription, and we're going to start out talking about transcription in prokaryotes, kind of like we did when we talked about DNA replication, and I told you that we worked that out in bacteria first because bacteria are much simpler beasties. They're unicellular, they have a single circular chromosome, and they have much, much fewer genes. So it's a much easier thing for us to observe. Additionally, you can grow them up very easily and they reproduce really rapidly. So for the same reasons, we are going to first look at how transcription behaves in prokaryotes, what happens, and then we're going to compare that to how that works in eukaryotes. So things like plants, animals, fungi, all that stuff. Eukaryotes, there we go. Okay, so we're going to compare and contrast how this thing occurs in prokaryotes and how it occurs in eukaryotes. So let's kind of go back and refresh our memories as to what we know about transcription thus far. You'll remember from Chapter 16 that transcription is, of course, copying DNA as a template into an RNA copy. So here we have a double helix of DNA. And this DNA has been unwound to form what is called a transcription bubble. And I'm just going to abbreviate trans bubble here. All right. This transcription bubble, remember, we're talking about DNA, so we have this part going from a 5 to a 3' direction. And here on our DNA, we're going from a 3 to a 5' direction, okay? Now notice that the RNA is only being copied on one strand of our 2 strands of DNA, isn't it? So we call this strand, and this should be reviewed from last chapter, we call this the template strand, which makes this one that we're not using up here, of course, the non-template strand. But I told you in our last lecture that there's some additional terminology that people frequently use to refer to these 2 strands. Sorry, drinking some coffee. So in addition to calling this the non-template strand, you will also see this called the sense strand. That's very common. And then this would be called the anti-sense strand. Your textbook also refers to the non-template strand as the coding strand. Now, oftentimes people find these 2 sets of terms really, really confusing. It feels a little counterintuitive. So let's kind of look at the rationale for why this would be called the sense or the coding strand when it's not even the one we're using, okay? Let's add some bases to our DNA. Let's just say that we have an A, a C, a T, and a G on this non-template strand of DNA. Now let's focus down here on our template strand of DNA. If we have an A, a C, a T, and a G on our non-template strand, then our template strand, the complementary bases that would correlate would be a T, a G, an A, and a C on the DNA. Now let's look at what that looks like in the RNA, and I'm just going to do that in a different color. Okay, the RNA that would correspond to this right here on the template strand would be an A, a C, a U, and a G. All right, let's look at this guy and let's compare it to our non-template strand. Hopefully what you immediately notice is that these are essentially the same. The only difference is, of course, that in RNA you always have U instead of T, right? You always use uracil instead of thymine with RNA. But that substitution aside, you can see that this sequence is identical to this sequence. So the reason that people call the non-template strand the sense strand or the coding strand is that if, as a molecular biologist, you're reading this sequence on the non-template strand, you know that you're reading a sequence that is exactly the same as the messenger RNA that is being translated later on into protein. So molecular biologists, we typically look at genes using this sequence right here, even though this isn't the strand that's being turned into mRNA, but the reason that we do that is because the sequence is identical to the mRNA. Okay, other things that I want you to notice, I'm going to just erase some of my stuff here because I want you to be able to see. I also wanted you to notice on this RNA strand, let me go back to black here. I wanted you to notice the template strand here is going from a 3' to a 5' direction. Notice that your RNA is being transcribed in an anti-parallel fashion. This new RNA strand that's being copied here is going from the 5' to the 3'. See this little arrow here? It's going from the 5' to the 3' direction. So just like DNA, RNA is made in a unidirectional fashion, going from the 5' to the 3' direction. Now, why is that? Well, let's go ahead. We're going to take this image in our next slide and we're going to turn it on its head because I sometimes think that's a little easier to envision or to conceptualize. So here's that same image, and hopefully you can see all I've done is I've turned it upside down. So this is our original transcription bubble here. Here is our growing mRNA strand that's being copied from the template strand, and we've magnified that here. So let's think about why RNA has to grow from the 5' to the 3' direction. Let's draw a nucleotide like we have 4 million times in this course. Here we have a base. I'm just going to put the letter B for a base. Here we're going to have an extra carbon and then a phosphate group. Now we're dealing with RNA, so this base I'm going to draw a little differently than I have been when I draw a DNA nucleotide. Remember, this is an RNA nucleotide. So that means that our central sugar right here is ribose and not deoxyribose. And if you remember from early in the course, ribose has an OH group on this 2' carbon. Deoxyribose lacks that. You would put an X through that if you were talking about DNA. That's why it's called deoxyribose. It lacks an oxygen. Now let's focus in on this guy here. This OH group that's on our number 3 carbon is the same in both ribose and deoxyribose. And this guy, as I've said before, is what I like to call the parking spot for the very next nucleotide that you would add in a growing RNA chain. So let's draw ourselves a second nucleotide that we're going to add here. Now I'm going to draw it just a wee bit differently and you'll see why. I'm going to draw 3 phosphates and then a carbon and then the rest of our sugar here with a base, OH, OH. Okay. And let's get rid of some of the stuff in here just to make sure that you guys don't get confused. But so what we're going to do is an enzyme is going to come along and it's going to take off this OH and it's going to grab a hydrogen from here. This is going to leave as water. And when that happens, it's going to weld these 2 nucleotides together. Now notice that I drew 1, 2, 3 nucleotides. This action of welding these 2 molecules together requires energy. And remember that energy is present in phosphate bonds. So if you break off these 2 phosphates, it provides the energy for an enzyme to weld together these 2 right here, okay? So maybe you remember that in DNA replication, we had what we called DNTPs. Those were deoxynucleotide triphosphates, and they provided the energy to zip up the sugar phosphate backbone in DNA. Today, we're talking about RNA. RNA has RNTPs. These ribose nucleotide triphosphates provide the energy to polymerize or add monomers to our RNA chain in this fashion. Okay, so that's what I'm showing you here, is that we have a growing RNA molecule, right? We've got a phosphate, sugar, phosphate, sugar, phosphate, sugar, and here is a new one being made. You can see we have 1, 2, 3 phosphates. We're going to build a bond here, and the energy from that is going to come from breaking these 2 phosphates off. Notice though that the reason we're growing from a 5' to a 3' direction is because you can only add at this 3' OH group carbon here. You can only add a new nucleotide here, and then you would only add another new nucleotide at another OH group down there. So just like DNA, RNA is built in a 5' to 3' direction. Just like DNA, we have an enzyme that links these guys up, so it's going to create this reaction here, or you can think about it as creating these bonds here. These bonds, if you recall, are called phosphodiester bonds, just like in DNA synthesis. Okay. And the enzyme that builds these bonds and makes this new chain is called RNA polymerase. And that should sound very familiar to you because you'll remember that when we were making DNA, we used a molecule called DNA polymerase. So essentially, transcription functions very much like DNA synthesis, but we're using different nucleotides and a different enzyme. However, we're still growing in the same direction. We're still matching up a new strand with an old strand. It's very, very similar. The last thing, though, that I do want to point out to you as a difference is you'll notice that this transcription bubble, we've got a non-template strand and a template strand, and here's our new RNA. Remember that this RNA is going to be single-stranded. Once we've copied it, it's just going to dissociate and float away to become a message that we then turn into protein. What will then happen is that these strands will re-anneal, which is a fancy word for saying they're going to stick back together like magnets, okay? So this is reusable, isn't it? Hold on, I wanted to go back here. Okay, so this is reusable, and this guy just floats off to become the message. And I'm trying to remember why I included this slide. I think I just included it to show you how similar DNA synthesis is to RNA synthesis. Okay, now here is an example of a final exam question I might ask. If I didn't have this labeled for you, how could you know that this was a growing molecule of DNA instead of a growing molecule of RNA? Think about that. And if you can answer that question, good for you. All right. Now, in thinking about how we're going to transcribe RNA, we have a little bit of a problem, okay? And that problem, let me actually insert a new blank slide. That problem is this. When we're copying DNA, hold on, I can't do two things at once, layout, blank. When we are copying DNA, we're starting at the beginning and we're ending at the end. We're copying all of the DNA that exists in the nucleus. So DNA polymerase can just start at the beginning and it can end at the end if you're talking about a bacteria. But if we're talking about transcription, we're honing in on a single gene, right? You're not necessarily transcribing every single gene on the DNA. You're only copying the ones that you need to turn into protein. So for example, later on, we're going to talk about a gene in bacteria that can digest a sugar called lactose. You only need to turn that gene on if lactose is present in the environment. If lactose is absent, then you wouldn't want to turn that gene on and make a protein capable of digesting lactose because that's wasteful. That wastes energy in the cell if you don't need it. So RNA is only going to be copied from the genes that the cell needs at the time. That means that we have to figure out a way to tell RNA polymerase where to start copying and where to end copying. So we need to figure out, come on, here we go, where to start copying our RNA and where to finish copying our RNA. Okay, so let's think about that by drawing ourselves a single gene. And I'm going to draw it a little differently, okay? In this example, I'm going to draw you a single piece of the DNA like a block. Understand that this DNA is, of course, double-stranded. So I'm going to draw a little line down the middle. So you have a 5 to 3 prime strand on top and a 3 to 5 prime strand on the bottom. But in thinking about the structure of a gene, you can kind of divide it up into different regions, okay? The first region that I want to draw your attention to is something called the promoter. This part is not transcribed, okay? It's going to be important in getting the process started, okay? And then you have a coding region down here. Now notice I drew a line right here to sort of delineate where the promoter ends and the coding region begins. And where that happens is called the transcription start site. So I'm just going to abbreviate trans start site. Now, as we move away from this transcription start site, we're going to say that everything, all the nucleotides, as we move in this direction, we're going to say that they are downstream of this transcription start site. And let's say that we wanted to talk about specific nucleotides along the way, specific A's or C's or T's or G's, because let's say we were looking at this sequence, we would number our nucleotides starting with +1 and ending with plus however many nucleotides there were in this gene, okay? If we're talking about the promoter, all this stuff in this direction from the transcription start site is called upstream. And you would delineate that stuff with negative numbers. So if we wanted to talk about specific sequences occurring upstream from the start site, we would use negative numbers, okay? So why am I telling you all this? Well, it turns out that in order to get this party started, in order to signal where the beginning of a gene is, the promoter has a couple of specific sequences, okay? And I'm going to draw here. It has a sequence in two places, one at the -10 position, and that sequence is something like TATAAT, I think. I'll have to go back and check my notes. And then there's another sequence at the -35 position, and I have that written for you somewhere. Let me see here. Ah, here it is. TT, let's see here. Yeah, TTGACA. All right. Let me go back. Here, okay. TTAGCA, or whatever I said it was. You don't have to memorize that. I'm simply pointing out to you that there's a little region here that has a very specific sequence of nucleotides, and that region begins at the -10 position. And there's a sequence here with a very specific set of nucleotides that begins at the -35 position. Now, why is that important? Well, it turns out that there is a protein, and I'm going to draw it in orange here, it has regions, amino acids on the protein that can bind to those sequences here, to these nucleotides here, and it can bind to the nucleotides here. So I'm going to draw it like this to kind of show you guys. There's a protein that is bound to our promoter, okay? This protein is called a sigma factor. Okay, this sigma factor is capable of recognizing this starting region, this promoter region, and the reason it can recognize it is because the amino acids on the sigma factor bind nicely to this sequence of nucleotides and the -35 sequence of nucleotides. Now, this sigma factor isn't all by itself. It, in turn, binds to RNA polymerase. So here's our RNA polymerase. And I'm going to color it in real quick, just so that y'all can see it. This is RNA polymerase here. So these two things are kind of bound together, but it's the sigma factor, the sigma protein, that is able to recognize the promoter by binding to those sequences. Now, this RNA polymerase, let me label this for you, and I'm going to abbreviate RNA whole. This combination of RNA polymerase plus a sigma factor is called the holoenzyme, which is pretty easy to remember. This is the whole thing, okay? So this mechanism is how we recognize where to begin transcription. Let's look at it in your book's diagram. So here we have a more accurate rendering of your DNA molecule with your sigma factor here and your RNA polymerase here, okay? This RNA polymerase by itself is called the core enzyme. And then remember, when it exists with the sigma factor, it is the holoenzyme. Now, I think that this is confusing to look at, so let's look at it in a diagrammatic fashion. You can see that here is our promoter of our gene, and it's bound up by sigma factor, which you guys can see in gray. Let me just change colors here to make this easier to see. So there's your sigma factor in gray, okay? Sigma factor is attached to RNA polymerase. So what sigma factor has done for us is located where to begin copying. So it's located the promoter of the gene, all right? Now, what's going to happen next is what is called the first phase of transcription. The first phase, and there will be 3, is called initiation. So what happens in initiation is you have to first locate the promoter, and that is what we have done. We're going to begin our transcription by locating the promoter of the particular gene that you are interested in transcribing as a cell, okay? And that's what's being pictured for you here. So sigma has bound the promoter. The second part of initiation, oh, let me do this. The second part of initiation is that the holoenzyme it moves through the promoter. Okay, so what do I mean by that? Holy smokes, I'm trying to write too fast and my tablet does not like it. So here you can see that they've got the promoter in this blue and purple color, right? And this whole thing has moved through the promoter, and the coding region is downstream in red. So what's happened, and maybe if I do this, let me just make this a little bit bigger for you. Well, let me, yeah. So what has happened here is that you can see this guy's moved through the promoter. We're now getting ready to start copying our DNA. Notice that RNA polymerase has done a couple of neat tricks. It has opened up the helix of DNA all by itself. In DNA replication, we needed a separate enzyme to pull apart the 2 strands of DNA. You might recall that that enzyme was called helicase. RNA polymerase does not need helicase. RNA polymerase can open up the DNA strand all by itself, okay? So after the holoenzyme moves through the promoter and opens up this helix, it's going to start transcription, but we're going to hold on to that for a second. The next thing I want you to notice is once it has moved through the promoter and it's getting ready to start copying this DNA, sigma pops off. Notice that sigma is here and it is not here, okay? So the next thing that happens in initiation, we start out, we locate the promoter, then the holoenzyme moves through the promoter, and then sigma pops off, okay? It goes away. And the thing that I wanted you to notice about this step here, remember, is that no helicase was needed simply because this guy could open up that helix all by itself, okay? We'll put RNA pol opens the helix. So what I've described for you here is how we're going to start transcription, how we start copying the gene, okay? The next phase of transcription is pretty simple, okay? It is called elongation, okay? So that's the second step, elongation. And in elongation, all that's happening is your RNA polymerase, let me make this a little bigger for you. Your RNA polymerase is moving along this strand of DNA. And you can see that as it goes along, it's opening up a transcription bubble. It's copying, it's adding nucleotides at complementary sites and then welding that backbone together. As it makes this new RNA molecule, what I hope you can see is that the new molecule trails off of the DNA. This is the original DNA here. Your new RNA molecules trails off while this continues downstream, okay? So it just elongates, elongation just elongates the mRNA, elongated. And that will continue until you get to a region of the gene. Let me move this guy up, oops. Here's what I wanted to do. Until you get, come on little guy, to a region of the gene called the termination signal, okay? The termination signal is down in this region. So let me draw you a little picture. This is our gene. Here is our promoter. Here is our coding bit. And at the very end is a sequence called the termination signal. And what the termination signal does is it codes for a sequence of RNA that I hope you can see what's happening here. This is a single strand because RNA is single-stranded. But notice that here, this single strand is starting to collapse upon itself because there are some complementary, let me clean this up, because there are some complementary base pairs, complementary bases. So this sequence here creates a situation where we get a hairpin because these things, the single strand starts to like connect to itself. When that happens, this hairpin creation causes the growing RNA molecule to dissociate from RNA polymerase and it stops transcription, okay? So this is called termination of transcription. It's how you stop it, okay? So transcription altogether occurs in 3 steps, and let me just write those at the top here for you in a different color. The 3 overall steps of transcription are initiation, then you have elongation, and finally, you have termination. Those are your 3 steps. Let's go back and look here. We have initiation, where you have the sigma factor way up there, recognizing the promoter. Then we move through the promoter and we begin transcription, which is the beginning of elongation. So this is initiation here. Here we have begun elongation. We're continuing in elongation. Then moving back down, here we form a hairpin, so we're beginning termination and we finish with termination, okay? Let's visualize this using a little animation that I have for you. This is the link in case you want to view it by yourself, but I'm going to go ahead and show it to you here. There's no sound, so I'm going to talk you through it. So here we're going to look at a little E. coli, and I like that it shows you, here's the bacterial cell. I don't want that. Okay, and then you can see we've got the cell wall, nucleoid region. It's showing you all the various places. Okay, we're going to zoom in on the DNA in the nucleoid region, and it's showing you the promoter of the gene that we're going to be transcribing. Here comes RNA polymerase that's already bound to the sigma factor, okay? And it is going to recognize that promoter, okay? They call that a closed complex, don't worry about that. But essentially, sigma has been able to recognize that region of the DNA. All right, moving on. It's opening up the helix. Transcription bubble is being formed. It's showing you the template and non-template strand. Now, it's cleared the promoter. Let me stop right there. We've finished initiation. We've cleared the promoter. We've opened the helix. Sigma has bound. The one thing that this animation doesn't clearly demonstrate is that sigma pops off at this point. At this point, sigma goes away and RNA polymerase is operating all by itself. All right, now we're at the transcription start site, +1. We're going to start adding nucleotides to a growing mRNA chain. So now we're in the elongation phase, okay? And we're gonna see that this moves through. As it moves through, can you see that we have an mRNA strand that is growing and growing and growing? Therefore, we call it elongation. Ignore the ribosome. We're going to talk about that in a second when we talk about translation. All right, growing, growing, growing until we see that a hairpin forms because of a termination sequence. When that hairpin forms, the mRNA dissociates from the RNA polymerase and we end our transcription, okay? If you watch this video on your own, you'll see that there's a second mechanism for termination in bacteria called row-dependent termination, but we're not going to talk about that in this class, okay? All right, let me go back here. Now let's focus in a little bit on how transcription in eukaryotes differs from transcription that we saw in prokaryotes. So what I just showed you was all to do with prokaryotes. In other words, bacteria. That's where we learned about all this stuff, was in bacteria. But now I want to talk to you about eukaryotes. Now I want to caution you guys about something. On your last exam, I asked you about eukaryotic extracellular matrices, and about 90% of you told me about animal extracellular matrices. So remember when we're talking about eukaryotes that we're not just talking about animal cells. We're talking about plant cells. We're talking about fungal cells. We're talking about any kind of unicellular organism, like maybe an amoeba, that has a nucleus and membrane-bound organelles. When I talk to you about eukaryotes, I'm not just talking about animals. I'm talking about any type of cell that has a nucleus and has membrane-bound organelles. Remember that eukaryotes are more complex than prokaryotes. Bacteria have a single circular chromosome, whereas eukaryotes, if you remember from like meiosis and mitosis, you've got a nucleus and you're going to have multiple linear chromosomes, okay? And I'm drawing little homologous pairs here with centromeres. And depending on the type of organism you have, you might have many different homologous pairs, okay? That means that eukaryotes have a lot more genetic material, okay? They've got more genes. Therefore, they have more DNA, and their genes are organized differently, okay? So these genes are organized differently than bacterial genes. So let's talk a little bit about that. And let's also talk about how transcription differs in eukaryotes than it does in prokaryotes. So this is the organization of a eukaryotic gene, and I'm going to talk extensively about that in just a second. But the first thing that I want you to know that differs between eukaryotes and prokaryotes is it has to do with RNA polymerases, okay? The RNA polymerase that I described for you in prokaryotes was a rather all-purpose enzyme, okay? It transcribed DNA into RNA, and it wasn't very careful or didn't care about what the purpose of that RNA was. But in eukaryotes, we actually have 3 different RNA polymerases: RNA polymerase 1, 2, and 3. And each of these RNA polymerases is responsible for transcribing a different kind of RNA. RNA polymerase 1 transcribes something called rRNA, which I'm going to tell you about in just a second. RNA polymerase 2 transcribes mRNA. And RNA polymerase 3 transcribes something called tRNA, which we're going to talk about extensively in our lecture 2 for this chapter. So what are these 3 types of RNA? rRNA stands for ribosomal RNA. So ribosomes, maybe you'll remember, and we're going to talk more about, but they are constructed of 2 different parts. Ribosomes have both a protein component and they also have some RNA, okay? Together, this protein and RNA gets all tangled up into a ball, and this ball has a very particular shape that is the appropriate shape for synthesizing new proteins. Remember, the job of a ribosome is to make new proteins. So, but this RNA here that makes up the structure of a ribosome, it stays RNA its whole life. It is not translated into protein, okay? Messenger RNA is. Messenger RNA, mRNA stands for messenger. And messenger RNA is turned into protein. So it essentially is the stuff that we're interested in this chapter. It gets copied from the DNA and then gets translated into protein. The last one that is transcribed by RNA polymerase 3 is called transfer RNA. The job of transfer RNA, we're going to see later, is that it connects amino acids with the mRNA, okay? And I will show you exactly how that happens in part 2 of the lecture. So the thing to know is that RNA polymerase 1 transcribes ribosomal RNA. RNA polymerase 2 transcribes messenger RNA. RNA polymerase 3 transcribes transfer RNA. This one and this one, I'll circle them. These 2 are not translated into protein. They stay RNA their entire lives. However, messenger RNA is translated, okay? Because that's what gets turned into protein. All right, so that's the first thing that you need to know that differs between eukaryotes and prokaryotes. Now, the next thing that you need to know has to do with the organization of eukaryotic genes. They are organized a little bit differently than prokaryotic genes are. So let's take and break down this structure into our own drawing to help us understand that. So we're going to insert a new slide. Let me just really quickly make that a blank slide. All right, so just like before, I'm going to draw for you a gene, and I'm going to draw it as a box, okay? I'm going to divide it in 2 so that you understand that we have 2 strands. Just like in bacteria, we're going to have a promoter, okay? But this promoter is going to be way more complex in eukaryotes than it is in prokaryotes, okay? And I will show you that in a moment. It has, instead of just having, one single region with a couple of different sequences, we have a sequence upstream here at the -30 position. It's called a TATA sequence because it's TATA. And that's pretty consistent among eukaryotic promoters, but it turns out there's a whole slew of additional sequences and other regions of this promoter. There's upstream regions called enhancers that can occur 1,000 base pairs upstream. This is a very complicated system in eukaryotes. All right. The other thing about eukaryotes is that we have a region downstream here after the transcription start site. So here's our transcription start site. We have a region that is transcribed, but it will not be translated. So we call this the UTR, for untranslated region. It's copied into mRNA, but it is not translated into protein. So UTR stands for untranslated region. Okay, then you have this whole region that will eventually be translated, and it is flanked by another UTR, or untranslated region. So we have what we call a 5' UTR and a 3' UTR. And then we have a termination sequence, but this termination sequence, I'm going to color it a different color so we don't get confused. Let's see here, let me go to draw. So I'll make this orange. This termination sequence, we give it a different name. We call it a polyadenylation signal. And you'll see why we call it that a little later. But essentially, this termination sequence signals an enzyme to cut the mRNA transcript at this point, okay? So we have this structure. The other thing that I'm going to go ahead and tell you about now, but we're going to talk about in a little bit, is that this region here that will eventually make up the coding region, it's organized very strangely into these chunks that we're going to call introns and exons. So we're going to call this exon 1, and this is going to be intron 1, and exon 1, or sorry, exon 2, sorry, and intron 2. And here we'll have exon 3. Now, what you need to know about these introns and exons is that this exon here, exons are excellent. They actually code for protein. So I'm going to write code and code. Oops. Come on, little guy. There we go. And code. All right. These introns are non-coding. Oops. Doing a bad job here. They are non-coding. They're nonsense DNA. So we're going to have some issues when we do some copying because we have these weird regions of coding DNA in our exons that are then interspersed with regions of non-coding DNA, which are called introns. I always remember that exons begin with X. Exons are excellent. We use those guys. They are coding. Introns, I always say they're insignificant. Because they are non-coding, okay? So the structure of a eukaryotic gene is going to pose some problems for us that we're going to take care of by processing our mRNA transcript much differently than we processed our mRNA transcript in bacteria. So that's where we'll pick up with lecture 2, and then we'll move on to translation. I'll talk to you soon. Bye-bye.